Good morning and welcome to Frontiers in Oncology. Well, it's my pleasure to welcome Dr. Kimryn Rathmell back to Stanford. Dr. Rathmell is the chair of medicine at Vanderbilt University Medical Center. She assumed this position just this past year. She was formerly the director of the Hematology Oncology division there. And she's also the Cornelius Abernathy Craig Chair. Dr. Rathmell was MSTP at Stanford, where she did her PhD in biophysics, and her PhD advisor was Dr. Gil Chu. She did internal medicine residency training at the University of Chicago and Penn, and oncology training at Penn. She was a faculty member at UNC before moving to Vanderbilt. She's a physician scientist, and she has been actively involved in leading several cancer genome atlas projects. She is an expert in the biology and clinical care of renal cell carcinoma, particularly the clear cell variant. And she has been interested in dissecting the hypoxia pathways that are dysregulated in clear cell in the kidney. So it is our great pleasure to welcome back Dr. Rathmell to Stanford. Unfortunately, it's not in person, but we're very happy to have you visit with us today and be our speaker at Frontiers in Oncology. Just a quick note to the audience, please write your questions at any time in the Q&A box below your screen. And please join me in welcoming Dr. Rathmell.
All right, great. Well, thank you for the kind introduction. I'm really happy to be visiting. I wish I was visiting in person. We're having a nice snowstorm here in Nashville. And it's really good to see a lot of friends in the participant list. I see a bunch of people from my lab too. So I'm being held to a very high standard. So we'll let them answer the questions, or hopefully they won't ask me too hard ones. Let's see, we could go. Okay, just a couple of conflicts. None of them will be relevant for today. Okay, so my lab studies kidney cancer, as you've heard, and we've taken a pretty broad approach. We're really just interested in understanding this beast. And so I put this slide up there partly to let you know that we are tackling this from a lot of different angles and to help understand why I'm really giving two talks today. The title might lead you to think that we'll understand a link between genomic instability and the tumor microenvironment. In fact, we won't. We study both of those things, and I'm going to have something both for the immunologist and the genome scientists in the audience. So kidney cancer starts in the kidney, and so I always start here. It starts, clear cell renal cell carcinoma starts in the proximal tubule, and it's important to remember that this is a really interesting place that's got an environment that's loaded with gradients of glucose, oxygen, and salts. And so it's a really interesting environment to start a tumor. The primary driver of clear cell renal cell carcinoma is a mutation in this gene. Hopefully you can see my arrow, called VHL, the von Hippel-Lindau gene. Loss of this leads to upregulation of a transcription factor called HIF. It turns on this very classic program of pseudohypoxia, and there's a strong metabolic component, and we've studied that within the context of clear cell renal cell carcinoma cells for years. But we work to understand a little bit better what genomic variations in the von Hippel-Lindau mutation do, what metabolic features are critical to the behavior of the tumor, other HIF targets beyond metabolic targets. There's a lot of influence in the microenvironment with angiogenesis signals, et cetera. And then, of course, there are mutations beyond VHL. And so we ask what other pathways cooperate as drivers and how this physiology now can play out in the tumor microenvironment. So the outline for the talk is that it's two talks. First is looking at metabolic competition and partitioning of nutrients in the tumor microenvironment. And then a second project that's looking at mechanisms of genomic instability related to SETD2. I'm giving both because I'm really excited about both talks or both projects right now. And because the second project brings me back home to some of my roots at Stanford. Okay, so first is the immunology talk. So kidney cancer is immune responsive, as you all well know. But like all of the other tumor types, we see some tumors respond really well. This example shown here is a patient of mine. Up top is pre and bottom is post. So we had a big renal tumor, multiple liver mets, was put on single agent nivolumab, and with time resolved all of his liver mets. His kidney tumor shrank down, in fact, to where that was all we could see. And we actually removed that, and it was loaded with immune cells. And we're now 3.5 years later, and he's disease-free. So why do some tumors respond like this and others don't? And what factors are influencing those immune cells? And can we influence the immune cells in the context of the tumor microenvironment so that we could have more patients like this? Those are the questions that led us here. So we had to ask, what do we know about glucose or metabolism in the kidney tumors? So not in kidney cells. After 18 years of working in the dish, we had to move into tumors. And what we know is, this is from an analysis of interstitial fluid and clear cell renal cell carcinomas. There's glucose there. There is lactate as well. There's other nutrients like glutamine. So the nutrients are present. We know that PET scans, glucose uptake PET scans, are problematic in kidney tumors. This has been an interesting problem for a long time. Here's an example of a big renal mass that's essentially PET negative, but there are others that are PET positive. When we do metabolic or molecular analysis of these tumors, those that are PET avid are actually more aggressive, carry more aggressive features. But we don't use PET very much in kidney cancer because it is variable. And so it's probably actually a biomarker, but not a good screening tool. Now, there are more cells in there, and we know that immune cells themselves are very highly metabolically regulated. Since moving to Vanderbilt, I've started to collaborate with Jeff Rathmell, who is a real leader in this field, and getting to know the immune cells on a different level. So T cell subsets, for example, if we look at the metabolome of these T cells, are all characteristically different from each other. So each requires a distinct metabolic program. And on a simplified level, this is where I am able to follow. Glutamine predominantly supports cells to shift toward a Th1 memory T cell set, whereas glucose is an important driver for the CTL T cell activation. Okay, so with that context, we decided to bring our labs together and deconstruct some of the metabolic properties of the tumor microenvironment. These are some of the people who are involved in this work. Katie Beckerman, who's now an assistant professor at Vanderbilt, along with technician Rachel Hungo. I have to give her a shout out. She's on the hunt for a graduate program. She's in at several top tier programs like Harvard and Hopkins, but is interviewing at Stanford, I think, in a week. So look out for her. And then 3 graduate students, 2 in particular have been leading the project that I'm going to focus on: Brad Reinfeld and Matt Madden, both MSTP students, and Melissa Wolf, who's also working in this project. So the 2 labs, I'm the kidney cancer lab and Jeff's the T cell lab, decided to see what's going on in the communication that happens between these 2. So this was Katie's project, was taking T cells out of a harvested clear cell renal cell carcinoma, comparing them to normal kidney cells as well as PBMCs. She makes a single cell suspension, isolates the TILs. And this slide tells a lot, which is that if you look at resting T cells, this is, I think, PBMCs from this patient, the CD4s and CD8s have a characteristic CyTOF signature. If you stimulate them with CD3, they have a characteristic activated signature. But the TILs that we take out of renal cell carcinomas are not characteristic of resting or activated. They actually don't activate very well. I don't show that here. They are sluggish, if anything, in their activation. They don't take up much glucose, and they occupy this sort of interesting middle ground. So they're a little bit distinct. When we look at them metabolically, they have very fragmented mitochondria, shown down here in the bottom left, both by immunofluorescence and EM. Those tiny mitochondria are hyperpolarized, shown up here in the upper left. And they have very high levels of mitochondrial ROS, less cytoplasmic ROS. And that high level of ROS is in spite of the fact that there are increased levels of reduced glutathione. So we know that they're metabolically in a state that's pretty unhappy. And the question is, can you change that? Or are they fixed in that state, or are they plastic? And three manipulations that we did show that they are actually plastic. If you look here, the gray is the TILs unstimulated. This is the normal level of stimulation we get with CD3. And we can boost that stimulation by bypassing this metabolic block. Here actually we're using a scavenger to get rid of the mitochondrial ROS. That allows them now to be stimulated. You can bypass the blockade. I told you they're not taking up glucose well, but you can feed them pyruvate. Now they activate quite nicely. And it's well known that if you block glutamine uptake, that cells will shift to uptake glucose. And if we block these cells or deprive them of glutamine with CB839, a glutamine synthase inhibitor, they'll now shift to taking up glucose and they'll now activate with CD3. So the question from there was, if the TILs are requiring glucose metabolism, what's rate limiting here? We've got cancer cells that use Warburg metabolism, are highly glycolytic in vitro. We have potentially nutrient competition happening in the tumors. There is glucose and it has to go somewhere. The T cells aren't using it, must be going to the cancer cells. And so we really started to deconstruct the tumor in a different level now. So the question was, what nutrients are cells actually taking up in the tumor? And so this is a nice cartoon that shows how complex these tumors are and the many different kinds of cells that exist in this environment. So what's in the tumor? We did some more interstitial fluid analysis. There's glucose. This is tumor versus normal. There's a glucose present, lactic acid, lactate's present, glutamine is present. We have a variety of murine subcutaneous models that we'll be looking at. Glucose, lactate, and glutamine are also present in these models. We can't look at micro niches, but in general, in the interstitial fluid, these metabolites are available. These graphs just show you the diversity of cell types, like the cartoon that we see in these model systems. These are three different models. The RENCA is a kidney cancer syngeneic model system. MC838 is a colon cancer model, and the polyoma middle T GEM model. Just there's a great diversity of immune cells that are present in it. That's the take home from the slide. So we take these GEM models, our mouse syngeneic and GEM models, and use F18 2-deoxyglucose like a normal PET scan, infuse them, take a picture like a normal PET scan. The mouse now has a radioactive tumor, and we remove the tumor and the spleen and sort over a magnet. We've done a variety of sorting mechanisms, but magnet seems to work the best into tumors, tumor infiltrating lymphocytes, myeloid cells, and we can do the same with the spleen, looking at T cells and others, and count the radioactivity. So where does the glucose go? So it's really an old school style experiment. And it turns out that it's actually in the hematopoietic cells that are taking up the glucose. This was a surprise that it was not so much the cancer cells. The FACS plots here are all just showing purity for people who want to see how pure these fractions were. But if you look down here at the bar graphs, we see glucose is taken up by the whole tumor, but proportionately, on a per cell level, is more in the CD45 positives than CD45 negative, which we are here calling the tumor cells. And we'll get more granular in just a minute. I'm showing this here in two different model systems, the RENCA and the MC38. And we've looked at a variety of other models, including GEM models and induced cancer models, all showing the same thing. This is the glucose is preferentially going to the CD45 set. So if we try to break this down again further, the FACS plots are all purity controls. And I'll draw your attention here for just a second. If we take the tumor and lay it on an autorad, there's glucose across the face of the tumor, so it's not at the outer edges. And the tumors themselves are highly infiltrated by CD4s and macrophage type cells, particularly this F480 set. So down here at the bar graphs, we look at spleen versus tumor. In that CD45, is it T cells? No, it's not T cells, it's other CD45s. Here again, now break those CD45s down, CD11B, which is a myeloid marker, and that's where we're finding this glucose being taken up. We also find it in GR1 positive cells and F480. So it's a myeloid macrophage particular subset of cells that are preferentially taking up glucose. are those cells actually more metabolically active? When we take these cells and put them on a seahorse, the most metabolically active set for both OCAR and ECAR is the CD45 positive F480 as compared to T cells or tumor cells. Now, if they're not necessarily competing, the question became whether or not this is a hardwired signal for these cells to preferentially choose glucose as their nutrient source. And we looked at and observed that phospho-S6 was highly expressed in both the cancer cells, CD45CA9, that's a marker of kidney cancer, and also here our CD11B, CD14 positive macrophage set. When we treat with mTOR inhibitor, rapamycin, you can see that the amount of glucose that's uptaken is reduced, consistent with a program of metabolic utilization that's driven by mTOR signaling. When we do gene expression studies, we've done RNA-seq and validated with a nanostring profile. Here, looking only at metabolism-related transcripts, you can see that, like I showed you at the very beginning of the T cell subsets, our immune cell sets are very selected by their metabolic transcripts. So you can separate based on metabolic transcripts, your MDSCs, your TAMs, CD8s, CD4s, and here's the tumor cells, the CD45 negatives. When we look at what transcripts are driving these separations, the tumor cells is largely amino acid metabolism and fatty acid metabolism, whereas your glucose metabolism is in the TAM set. So what particular genes, here's some just example genes of the TAM program that's driving glycolysis. Hexokinase 2 is selectively expressed in these cells, and that expression level is decreased when we treat with rapamycin here in the TAMs. CD98, on the other hand, is a cofactor that drives amino acid transport, and we had seen amino acid transporter as an important part of the tumor signature, and that's also affected by rapamycin signal. So if fatty acid and amino acid metabolism is skewed toward tumor cells, are they actually preferentially taking those nutrients up? And we see, in fact, that they are. So here we're looking at C16 Bodipi as a lipid that is preferentially being taken up by the CD45 negative. This is our tumor set as compared to TAMs, F480 lows, and CD3 positive cells. So lipid is preferentially going to the cancer cells, and glutamine too. So glutamine we can look at with an F18 glutamine PET scan. Here's another one of those autorad tumors showing the glutamine uptake across the face of the tumor, and which cells are taking up glutamine. Here it's not the CD45 positives, it's the CD45 negatives. Here it's the cancer cells. And as we had shown before, when you hit with rapamycin, we had seen that takes down CD98 and it reduces the F18 glutamine uptake. Now, the T cell world and others have shown a tight correlation between glutamine and glucose uptake. And if you shut down glutamine uptake, you'll preferentially shift to taking up glucose. And we were able to show that here in the whole animal. So treating animals with V9302, an inhibitor of ASCT2, a glutamine transporter. Here at the left is really a control. Looking at F18 glutamine uptake, it shuts it down, as it should, a good transporter. But what does it do to FDG uptake? FDG uptake is increased both in the CD11Bs, but also in the CD45 negative, the tumor cells, and in other CD45s as well. So we can shift this with manipulations that are in the whole animal level. So it's exciting, and I'll just put my bullets up before I run through them. The metabolism in the tumor and microenvironment is probably more than simple competition. We've shown that T cells show signs of exhaustion, but can be rescued metabolically, and that in vivo glucose uptake in tumors is mostly hematopoietic. And this is across like 6 different tumor models, and we've shown in some human settings. And while glutamine uptake and lipid uptake is more cancer cell specific, glucose competition in tumors is modest based on the amount of material that we see available, and FDG PET probably reflects inflammatory component as much as it does the cancer cells. and that nutrient utilization and metabolic program is driven by mTORC1 signaling and can be manipulated with glutamine inhibition. So that is story number 1. And I'll take questions at the end, just make sure I'm doing all right for time. And I apologize for burning through, but I really wanted to share both stories. So we're going to shift gears entirely so the immunologist can rest. And now it's the genetics folks. We're going to look at mechanisms of genomic instability related to SETD2 deficiency. So kidney cancer, I've told you, is driven by this VHL mutation. It has almost exclusively a 3p deletion. So there's VHL mutation and obligate loss of heterozygosity that leads to, as I told you, HIF stabilization, an angiogenic signature, and a metabolic signature. But there are secondary mutations as well. And those, it turns out, are almost exclusively in chromatin modifiers. So it's PBRM1 about 40% of the time, SETD2, which is a histone methyltransferase, 15%, BAP1, 10%. The really interesting thing about these is they're all co-localized on 3p. So if you look up here, this is the kind of classic paper from the TCGA index papers, clear cell renal cell carcinoma. So VHL mutation, very common, 3p deletion is almost 100%. And that 3p isn't just where VHL is, it's a whole segment of 3p, and it always encompasses all of VHL, SETD2, PBRM1, and BAP1. And so all of these tumor suppressors have obligate loss of heterozygosity. And so their mutations serve as secondary drivers to VHL. This is a summary of what they do, but I'm going to focus on just one. So SETD2 is a methyltransferase for histone H3 at lysine 36. So I'll use this term, H3K36 trimethylation, or H3K36ME3. That's its signature activity, is to drop that third methylation group on H3 lysine 36. It binds to RNA Pol2, and it's well known to move along actively transcribed genes. Along a transcribed gene, you'll see an increased amount of H3K36 trimethylation, and it's a marker of these actively transcribed genes. And a number of different reader molecules can see this trimethylation mark, like DNMT3B is one example. And so it's a marker that helps to guide DNA methylation, among many other things. So SETD2, when it's mutant in kidney cancer, leads to global loss of this mark, this H3K36 trimethylation mark. Two former MD PhD students in the lab, Catherine Fahey and Kate Hacker, did a lot of the work. I'm just going to show one tiny piece of their work, but showing that this is a non-redundant trimethylation mark. So if you knock out SETD2, you lose it in all the cells of the tumor at a global level, or at a gene level. Here, we're looking at metagene signature ChIP-seq for H3K36 trimethylation. Normally, it's present on the genes and increases across the gene body. And you can see when we knock it out, it's just gone. So it's gone. So we've been working in SETD2 now for 6 or 7 years, and sort of setting up the stage. We know now that SETD2 loss results in a widespread open chromatin phenotype. There is an increase in altered splicing. We know that SETD2 and SET2, the yeast homolog, behave a little bit differently. We used a mutation series to show that the cryptic initiation, so H3K36 trimethylation was originally thought to be a barrier to cryptic initiation based on work in yeast, but it turns out that's not true. That's actually repressed by dimethylation. So that took one thing off the plate of what H3K36 trimethylation might be important for. It's not important for cryptic initiation. SETD2 regulates transcriptional fidelity that facilitates cell cycle progression. That may be important, as we're going to get into some mitosis work in a second. It turns out haploinsufficiency, which, remember, 3p deletion is a canonical feature of clear cell renal cell carcinoma. So 15% will have SETD2 mutation and 3p deletion. 85% will just have 3p deletion. But that haploinsufficiency is sufficient to drive genomic instability. The loss sensitizes to PI3 kinase beta and AKT inhibition, and it has other targets. SETD2 can also methylate components of the cytoskeleton like alpha tubulin and actin. And I'm not going to talk about those much today, but happy to in the discussion because it may be important, but we're setting that aside for a minute. So renal cell carcinoma with this VHL mutation and then secondary chromatin modifiers wasn't thought to be one of these tumors that was very highly mutagenic. And in fact, if you look at tumor mutation burden, it tends to score on the lower side. But it has this really interesting feature that was recognized in about 2012. One group took one tumor and sequenced all across the tumor and the metastasis, and we're really surprised to find that the heterogeneity was intense. Almost every sample was distinct, and so the clonality was, there were some features that were at the core. They called them Truncal mutations. That's VHL-SETD2. But it just kind of got exploded from there. This was a pivotal paper. It was published in the New England Journal, and as I said, in 2012. And it turns out that there are some tumors like that, and not everyone is. So many tumors with in clear cell renal cell carcinoma will have a more classic clonal type of feature set if you sequence them multiply. But tumors with SETD2 mutations have this high heterogeneity phenotype. So there, it seems like a good setup for some genomic instability that is ongoing. When we knocked out SETD2 in a variety of different cell settings, we observed that SETD2 was inducing a chromosomal segregation error. And I used this picture that we published in a paper a couple of years ago because it shows almost all of the features that we see at mitosis. So we see bridging chromosomes, we see lagging chromosomes, and we see chunks of genetic material that are not associated with the spindle at all. So mitosis is clearly disrupted in these cells, and you can see if we quantify, roughly even proportions of lagging, bridging, and other mitotic errors. There are a lot of things to unpack in here, with lagging chromosomes having probably a feature due to improper microtubule chromosomal attachment. But we're going to focus on bridging chromosomes. So bridging chromosomes are thought to be caused by structural changes in chromatids, and the degree to which we saw bridging chromosomes really made us think that we needed to understand what was causing that bridge to persist. So one feature of bridging chromosomes is, the failed mitosis of these is a genomic instability mark of leaving behind micronuclei. Jean Chang was a postdoc in the lab who worked on this project and showed that, in fact, when we knock out SETD2, here we're using mouse embryo fibroblasts, whether we knock out both copies or only one copy, we see a massive increase in the accumulation of micronuclei, some of which will harbor a centromere, which may become important later. So you can see both micronuclei with and without centromeric material, but like 60% of the time, so lots of micronuclei. If you see this much micronuclei, then it must be present in the cancers too. So one side benefit of Project One that I talked about, getting all of these tumors from humans, is we have some people taking the T cells away, but we can look at the cancer cells, and looked at those cancer cells and found that if we took from normal kidney, we never found a micronuclei, but we had a very significant increase in the presence of micronuclei among tumor cells. This is just some examples here. So we're seeing micronuclei, we're seeing bridging and lagging chromosomes. I just also want to show you a picture. We also see aneuploidy and the accumulation of cells that have two nuclei, and that we think ties to this retained bridge. So in this little movie, I'm just going to show you that we have two cells that just completed cell division. The nuclei are covered here, and then underneath of this string is going to be where there's a bridge. Okay, so the bridge is gone, and these cells that just finished are hanging out. And how do you get binucleate? Well, they come back together because you get failed abscission. So we see this happen not at all infrequently. So not only do we see bridges, but we see that those bridges don't always even get resolved. Okay, so that's just fun to show. So Frank Mason, who's a research assistant professor in the lab, cell biologist who did his postdoc at MIT, has really taken this on to champion us and help understand what's going on with SETD2 and the formation of these bridging chromosomes and how they might be driving genomic instability. He works with Emily Kunlevong. She's a technician in the lab and just recently had her first acceptance to an MD PhD program, so we'll be an MD PhD. So first question. So here we have H3K36 trimethylation that we know happens on gene bodies, and there's really not any data of it being present anywhere else. But if we have such a profound impact on mitosis, we felt like we might need to look at the centromere. And so looking at, this is strands of DNA that are pulled out, you can see, we can mark the centromere with CREST, and H3K36 trimethylation is present at the centromere. So great. And if you co-IP or chip co-IP, you can see that H3K36 trimethylation also co-IPs with Senpai. So I need to tell you a little bit about Senpai because it's not on the tip of everyone's tongue, but it also helps to sort of understand some of the assays that we're going to do. So Senpai is an histone 3 variant, so it actually replaces histone H3. So you can't have the same molecule. Senpai isn't methylated. So in the area of the centromere, Senpai replaces many of the nucleosome histone H3s, and that's the mark of a centromere. There are additional marks we'll talk about in just a second, but we're going to look at Senpai often as our mark of a centromere. And Senpai is localized at the center of the centromere, and it's known that the nucleosomes flanking that, the pericentromeric region, are H3K36 dimethylated, and are thought to have a boundary setting mark for the rest of the heterochromatin that's outside of the centromere. H3K36 trimethylation really hadn't been looked at before. We just showed you that it's there. So it's present at the centromere. And in fact, it's present also at pericentric regions. So here I'm just showing you some spread metaphase chromosomes showing H3K36 trimethylation. HEC1 is a marker of the kinetochore, so that's only going to be present at the centromere. And here for this chromosome, you can see here's centromere region and H3K36 trimethylation flanking it. Of course, it's present across the length of the chromosome as well. It's also present at the very tips and appears to be present in telomeres. Before I move to the next, I'll just show when we knock out SETD2, it's gone. Okay, so we know that this mark really is SETD2 dependent. This is H3K36 trimethylation flanking the centromeric region. Here is another example, another cell line. We're staining the centromere here with CREST. You can see flanking areas, H3K36 trimethylation, also very present at the telomeres as well as across the length of the chromosomes. Again, knock it out and it's gone. And so this is our map of H3K36 trimethylation, highly focused in pericentric centromeric regions. What's it doing there? We're actively working on, and it's also present in telomeres. So what happens when you lose SETD2? When we lose this H3K36 mark, what happens to the centromere? So when we knock out SETD2, you lose H3K36. Here are just 3 different cell lines. The levels of Senpai actually increase globally. So just by Western blot, we see increased Senpai. And if we look at individual centromeres, the intensity signal is increased. So the centromere density of Senpai is increased. And you can find Senpai more broadly spread across the chromosome. So this is controls, and here's where we knocked out SETD2. So normally you see Senpai really, or CREST is what we're staining for here. Just at the centromere region. Mouse chromosomes have always have this skewed chromosome pattern. But when we knock out SETD2, you can see that it's spread across the chromosome, and there are foci that are as intense as centromere. In fact, so that you can't tell which one is the centromere and which one is the new extra, if we're assuming that we've just increased Senpai and disposited across the chromosome. A few more examples showing, centromere spreading across the chromosome compared to normal, and then a second. And it actually made us think, this is not just spreading and clustering deposition. Are these really neocentromeres that we've made, or are these actually dicentric chromosomes? Just a few more examples. Here's before knockout, after knockout, with lots of these 2 centromere looking chromosomes. So if we quantify how often this happens, we see about 60% acutely will have dicentric, of spreads will have a dicentric chromosome. Sometimes you can see more than one. There are also acentric chromosomes. They're not so much to look at because they're just little blobs of DNA without a centromere. But you see acentric and dicentric. I'm going to call them dicentrics, and I'll tell you why in a minute. It's not only happening in our mouse model. This is in human cells where we knock out SETD2, and you can see we get what appear to be dicentric chromosomes as well. Now, when we look at these dicentrics, or these multiple centromere chromosomes, we can ask whether they just have the proteins that mark as centromere, or do they actually have SEMP-B as well? You can fish for SEMP-B gene, or SEMP-B is another marker of a bona fide centromere. So do we see both marks? We are also going to look at HEC1 to see if the kinetochore starts to build, or do you only see one, suggesting that we just have a SEMP-A clustering effect? In fact, we see all of those effects. What we're going to show here, this is normal. The models are, we call B+. B+ means you get A and SEMP-A and SEMP-B, SEMP-A and SEMP-B at both foci. You might get both together and only SEMP-A, or you might get only SEMP-A. We can see all examples. So here's a BB+, here's a B+, B-, here's a B-, B-. So that's frustrating to see so many, but if you actually quantify it, it's actually mostly dicentric chromosomes. So this is why I've been calling them dicentrics. The great majority, 88% of them, will have both SEMP-A and SEMP-B at their at this centromere that we're observing, whether we look at it by immunofluorescence or fish marker. So we're going to just focus on dicentrics, even though it seems like a few other things happen along the way. I mentioned that we'll look at HEC1 as kinetochore assembly, and you can see that HEC1 can be present as well. So these are at least beginning to form spindles. Okay, and they're persistent. So it's not just one flush and those cells all die. We see initially with knockout about 60% of spreads. That drops down and stabilizes somewhere around 20% of spreads having a dicentric chromosome. Yeah, I think it's always important to go back and look at human tumors. So here's a human renal cell carcinoma cell line with 3p deletion. We've looked in normals and clear cell renal cell carcinomas, and we do see evidence of dicentrics as well in human tumors. So fresh from the patient human tumor. And the dicentric chromosomes do appear to contribute to bridging. And here looking at anaphase is an example of we're staining for crest and DNA that these dicentrics are probably contributing to the bridging as we suspected. Now, well, some of you may have noticed, I didn't draw attention to the fact that when we see our dicentrics, they tend to look a little bit different than the rest of the chromosomes. They are often larger, as in this example from a human experiment where we did HeLa SETD2 knockout, and they often appear quite symmetrical to our eyes. So the question was, are they comprised entirely of one chromosome? Or is this a fusion of chromosomes? Are they isodicentric or palindromic? And so the only way you can address that is with spectral karyotyping. So this is the mouse embryo fibroblasts induced with tamoxifen to knock out SETD2. So we get a big blush of dicentric chromosomes, and then they're painted. We do see, again, some examples of dicentrics that are intrachromosomally rearranged. But again, the great majority, 85%, don't have interchromosomal rearrangement. They are comprised of one chromosome. So they have one chromosome type with two centromeres. So what could be driving dicentric chromosomes in SETD2 mutant cells? So there are a lot of different possibilities. It could be double-strand break driven with chromosome fusion. Based on the fact that we see mostly isodicentric or one chromosome type present with the dicentrics, leans against this being double-strand break mediated. But I already showed you that H3K36 is present at telomeres. So could it be that our telomeres are also defective? And so we're seeing some telomeric fusion. We've done work to look at telomere attrition. That does not appear to be happening. The telomere length is very stable in these cells when we knock out SETD2, so probably not that. And and then finally, another option is this just neocentromere formation and inactivating the proper centromere? And I've already shown you that that we're seeing these aren't truly neocentromeres, so probably not. But we did G banding then, because we know it's one chromosome type, two centromeres. They look pretty palindromic to us, but G banding is the only way to really characterize this. And the isodicentrics are greatly outnumber dicentrics that are otherwise not otherwise specified. So we do see isodicentric. So these are palindromic dicentromeric chromosomes. So not neocentromeres. And the final is, I don't have the final answer on what's driving this exactly, but how do you get a palindromic palindromic chromosome with dicentric features and acentric chromosomal material? There are defects in double-strand break repair, taking me back to my old days of COO 7080 that could be driving this. But the model that we favor is actually a faulty template switch, which is a replication template switch mechanism. It's break independent that can drive a dicentric formation with acentric material. And we're actively trying to solve whether or not this is the driver. So I'm sorry that I don't have the final answer to that, but I really wanted to bring this story forward to chat about. So to summarize this, we see bridging chromosomes, micronuclei, and binucleation occurring with SETD2 loss. We see this in all of our cell line models, and we're seeing evidence of this in human tumor specimens. H3K36 trimethylation is present in pericentromeric regions along with dimethylation, and loss of that mark is associated with increased Senpai at the centromere, Senpai spreading across the chromosome arms, and acentric, dicentric, and palindromic chromosomes as the dominant genetic feature. We do see neocentromeres, but they're much less common than having a second bona fide centromere. And dicentrics that we find are mostly isodicentric palindromes. So in the big picture, we have renal cell carcinoma that develops in the setting of pseudohypoxia. The tumor microenvironment, back to the first project, involves a metabolically very distinct set of cell populations. And when we look at secondary drivers of our cells, they promoted genomic instability through bridging dicentric chromosomes. And with that, I'll end with some acknowledgements. The first part of the project for sure couldn't have been done without collaboration with Jeff Rathmell, and so it's great to have an immunologist in the house. I've tried to acknowledge everybody who was involved in these projects as we've gone through. This is the lab, past and present, and some of our important collaborators. And with that, I will stop and take questions.
Well, thank you, Kim. That was really exciting. I'm trying to turn my camera on, but
Oh,
the host is not allowing me to,
oh,
whoever the host is.
not me, hopefully.
No. Okay, great. Thank you, Maria. Maybe I'll get started. I was really interested and excited, especially by the genomic instability part, because it relates so closely to work that I had done in the past on telomere instability and how that generates dicentric chromosomes. And I wondered, in your MEFs,
Mm-hmm.
you have 15% of the fused chromosomes or dicentrics that are translocations, essentially, between two
Mm-hmm.
different chromosomes, and
Yep.
then the 85% are a single chromosome. And the structure of those is really interesting, and it almost looks like the chromosomes are fused, like the Q arms are fused to each other, what we used to call a QQ fusion, which is the opposite of what we saw with telomere shortening in the mouse, which was always a PP. So just for the audience, Kim referred to the unusual structure of mouse chromosomes. They have very, very short P arms, and so you almost can't see them, but you can see them if you do a telomere stain. Mm-hmm. And then almost all the genes are encoded on the long Q arm. So what you were showing looked like they could be, but they may not be mm-hmm. Q arm fusions. And I wonder, is that what you think is going on, or is it something else?
Well, so we have so I think if there's a fusion happening, it's a QQ fusion, and you're right, actually, for the 15%, those really look like that's where those fusions are occurring. And we did look at the telomeres of the P arms as well as the Q arms when we were trying to decide if this was a telomere shortening event. What I don't know is if that kind of fusion is also just happening in the interchromosomal, same chromosome fusing. We don't have a great way to address that yet, and so we are favoring this creation of a palindrome by template switch, because it happens so frequently that somehow there's never a break. It is sort of our model, but it may be that there are breaks and there's just some stochastic driver that we don't understand as to why they would choose an intermolecular fusion as opposed to nearest neighbor fusion.
I'd be curious to know if there's any telomere sequences at those joins. You've done some.
We've looked for that. They're not.
Not there.
Yeah.
So it's probably something else then.
Yeah.
It's very interesting, genomic instability phenotype.
Yeah.
Okay, there's a question in the box from Nathaniel Gray. He says, can you comment on the effects of ketogenic diets on immune infiltrates if you examine these?
Yeah, that's a really interesting question. We're in Nashville, right? So we have not looked at ketogenic diets, but we have some folks who are looking at obesity and high carbohydrate, high caloric diet. So we probably need to go the other way as well. Preliminarily, I can't say a lot. There's definitely an influence of obesity on the immune cell repertoire. What it's going to mean for the actual metabolism of those immune cells isn't really clear. But it's a really good question. I think shifting the animal model beyond like genotype to those kind of environmental influences, there has to be some effect, and I think it'll be fun to look into it.
Thank you. Alice Fan is saying, thank you for that terrific talk. Can you comment on the role of metabolic aberrations for detection and treatment of renal cell carcinoma? We were disappointed that the first glutaminase inhibitor studies were negative in RCC.
Yeah, no, I think that's a great question. And we were super disappointed when that data came out as well, because it really looked like you could manipulate both the immune cells and the tumor cells with the glutaminase inhibitor, or at least altering glutamine biology, at an organism level. First off, I think there are other inhibitors. So one glutaminase inhibitor doesn't mean that the whole model fails. We've worked with that glutaminase inhibitor, and as you saw the data I showed is actually from an ASCT2 inhibitor. and it may be that transport inhibitors are actually more valuable. So it may be the mechanism that we're using to try to target these metabolically could be important. I would love to be able to do glutamine PET scans, for detection. I think it would be really lovely to have that option to look both at, where is glucose going and where is glutamine going. I think we may get a more robust picture of what's tumor cell versus mixed tumor and inflammatory when we're doing a glucose uptake. And I think there are bunches of other PET tracers that are on the horizon.
Great. Someone you may have heard of named Gil Chu is asking a question. He says, thank you for your talk. You showed convincingly that SETD2 mutation leads to intratumoral heterogeneity and dicentric chromosomes. Do different subtumors have different dicentric chromosomes?
Yeah. No, yeah, you didn't miss anything. We have not done anything at that kind of level yet. The dicentric chromosome, so we don't know if the dicentric chromosome is going to persist or if it's going to be, a new dicentric emerging with subsequent rounds of replication every time. I think that's the way it'll likely look, that we're breaking chromosomes over and over again, and have a system that keeps making new dicentrics. That's my mental model of things. But I think, the clonality of these tumors is really hard to really wrap your head around. What's left to tell you what the original insult was when you're many mitoses later in the tumors is pretty hard. We're trying to do that, in a more controlled way in cell lines where we knock out SETD2. That's why we've started to look at persistence of dicentrics. But even there, we don't know if it's the same dicentric. And again, I suspect it would not be. I think it's just sort of a base level of dicentric that gets created.
I have a question that maybe is unanswerable, but I just wondered if SETD2 could be a rare BRCA syndrome variant. Is that ever inherited as a rare BRCA allele?
Yeah.
Given your data.
Yeah. So SETD2, first off, is showing up in lots more tumors. So it was originally identified in renal carcinoma, and a bunch of hematopoietic tumors actually have SETD2 mutations. So I think this is going to be important well beyond kidney cancer. That's just our wheelhouse. In terms of showing up in these genetic panels as a inherited driver, I don't know if we'll see that or not. Both NSD1 and NSD2, the dimethyl transferases, and SETD2 have been found in conditions that cause sort of gigantism, but not tumor. So there are human conditions of NSD1, NSD2. There are very few examples with SETD2, but at least one or two families mirrored what they saw with NSD1 and NSD2 mutations. That doesn't mean we won't find it when we start looking at broader panels of genes, but I don't think it's been seen yet as a hereditary risk factor.
Thank you. Adele Zhu is asking, could any genes that are transcriptionally activated by SETD2 be implicated in the genomic instability that you observe upon SETD2 knockout?
Yeah.
Good question.
Yeah, it's a really good question. So we originally started all this being sort of more transcription-y people, and it's really interesting. Although when we knock out SETD2, we can see pretty gross changes in chromatin organization, right? So if we do like fair-seq, ATAC-seq, you'll see big changes. We don't see very many changes in transcript. Actually, almost none. I mean, you have to look hard to see, what's really changing at a transcript level, other than, indicators that you've messed up mitosis a little bit. So it doesn't seem like it really changes the transcript signature at very substantial ways. which is really surprising to us. That's actually how we got started. We were looking for transcript effects and really not seeing any, and that's where we started looking at splice variants. So we did see an increase in kind of general splice variation, not one splice variant that could serve as a driver, more sort of lots of splicing error. It seems like the system suddenly just gets messier when you lose SETD2 in that way.
Great. Well, we're almost at the top of the hour. Any final questions for Dr. Rathmell? If not, I want to thank you for a really terrific seminar, Kim, and thanks for joining us back at Stanford. And we'd love to host you again back in person.
That would be great.
When the future allows. And best of luck to you in your science and your new exciting administrative roles as well. And thanks for joining us.
Yeah, thanks very much.
Okay, our next presentation for Frontiers in Oncology is Omar Abdel Wahab, and that's on March 2nd. And he is from Sloan Kettering and will discuss understanding and targeted altered RNA splicing in cancer. So please join us on March 2nd and have a great day. Thank you all. Thank you, Kim.
Thanks.
Bye-bye.